bacterial genetics, is the absolute foundation of modern biotechnology.
It's the toolkit.
It's the entire toolkit. I mean, every major medical breakthrough in the last 40 years, from insulin production to the mRNA vaccines we've all heard so much about recently, they all rely on the exact mechanisms we are going to discuss today. We're essentially decoding three main things here. First, how bacteria store their identity, their DNA.
Yeah.
Second, how they actually change that identity through mutation. And third,
is the crazy part.
right, the crazy part is how humans have learned to break into that system and just hijack it for our own engineering purposes.
It's basically a heist story. We are breaking into the vault of life. But before we get to the actual heist, we really have to understand the machine we're looking at. And the thing that just blew my mind in the reading is just how unbelievably fast and efficient bacterial genetics are compared to us. I mean, humans are great, we have big brains, but biologically, we're kind of slow.
Glacial. We are glacial compared to them. Bacteria are built entirely for speed. They are these streamlined survival machines. And to understand that speed, we really have to start at the bottom with the blueprint, the DNA.
Right. And the source material starts off with a distinction that I think is, well, it's really important because people mix these two terms up all the time. The difference between the genotype and the phenotype.
It is a totally critical distinction to make right up front. Think of it like this. The genotype is the code on the hard drive. It is the specific physical sequence of DNA nucleotides hidden deep inside the cell. It's the potential of what the cell could be.
Okay, so the genotype is the raw file.
Yes. And the phenotype is what actually appears on your monitor. It is the physical, observable expression of that hidden code. It's the real-world result. So let's say a bacterium has a gene in its DNA that codes for an enzyme that can break down penicillin. That gene, that exact string of DNA letters, that is the genotype.
But the fact that the bacterium actually survives when you take antibiotics.
That is the phenotype, exactly, the observable trait. And the reason this matters so much for our deep dive is that you can have changes in the genotype, silent mutations in the underlying code that don't change the phenotype at all. The code changes, but the screen looks exactly the same.
But evolution can only see the screen, though, right? It can't read the hard drive.
That's a fantastic way to put it. Natural selection acts strictly on the phenotype on whether you live or die in a given environment. It doesn't care what your hidden code looks like, as long as the end result works.
Now, actually proving that this connection existed, that there was a real physical molecule carrying this invisible information, was a massive scientific detective story. The notes mention this thing called Griffith's experiment from the 1930s.
Oh, this is the ghost story of genetics.
It really is. So set the scene for us here. It's 1928, Frederick Griffith. What exactly is he looking at in his lab?
So he's working with Streptococcus pneumonia. This is the bacteria that causes pneumonia, which, remember, was a massive, terrifying killer at the time. And he's looking at two completely different strains of this bug under his microscope.
One is called smooth and one is called rough.
Right. The smooth strain looks shiny under the scope because it has this capsule, a thick, protective slime layer on the outside. This capsule is basically armor. It completely hides the bacteria from the host's immune system. So this strain is virulent, it's deadly. If you inject it into a mouse, the mouse dies very quickly.
Okay. And the rough strain?
It has no capsule, it has no armor. It just looks rough and bumpy under the microscope. If you inject this one into a mouse, the mouse's immune system spots it immediately, attacks it, and destroys it. The mouse lives.
So far, this is just basic pathology. Deadly bug kills the mouse, the wimpy bug doesn't.
Correct. Nothing groundbreaking yet. But then Griffith does something strange. He takes the deadly smooth bacteria, puts them in a tube, and boils them. He kills them completely with heat.
So they are super dead.
Dead as a doornail. He injects these dead, boiled bacteria into a new mouse. And predictably, the mouse lives. Dead bacteria can't kill you.
Okay. But here comes the twist.
The remix. He takes a mixture. He mixes the deadly bacteria with the living, harmless bacteria.
So logically speaking, you just have corpses and wimps. Nothing in that syringe should be able to hurt the mouse.
That's exactly what logic dictates. But when he injected this specific mixture into the mouse died.
That is so creepy.
It gets even worse. When Griffith did an autopsy on that dead mouse, he didn't find the harmless, rough bacteria inside it. He found living, reproducing, smooth bacteria. The harmless bacteria had somehow resurrected the deadly trait from the literal corpses of the killed bacteria.
It's like walking past a graveyard and suddenly knowing how to do karate just because a karate master is buried there.
That is a brilliant analogy. And the thing is, Griffith had no idea how it happened. He couldn't explain the mechanism. He just called it the transforming principle. Something invisible was being passed from the dead cells to the live ones.
And for a really long time, scientists thought that invisible thing was protein, right?
They did. Because proteins are vastly complex, they have lots of different building blocks. DNA, on the other hand, seemed way too simple. It's just 4 chemical letters repeated over and over. They thought, how could this boring, simple sugar string possibly carry the complex code of life? It wasn't until the 1940s that researchers Avery, McLeod, and McCarty finally did the hard chemistry and proved that the transforming principle was, in fact, DNA.
So the dead bacteria's DNA was just still perfectly intact, floating around in that boiled soup.
And the living bacteria just picked it up, read it, and said, oh, so this is how you build the slime armor. And they just built it.
Which perfectly brings us to the molecule itself, the famous double helix. I mean, we've all seen the pictures, Watson and Crick. But looking at the source material, there are some real nuts and bolts here that explain why it works the way it does. It's not just shaped like a spiral staircase because it looks cool.
No, the structure is strictly functional.
Yeah.
Every part has a purpose. You have a sugar phosphate backbone. That's the sturdy handrail of the staircase. And then you have the rungs connecting the 2 sides, which are the nitrogenous bases.
Adenine, thymine, guanine, and cytosine. A, T, G, and C.
Exactly. And they follow the absolute golden rule of genetics. A always pairs with T, and G always pairs with C.
But there's a specific detail here about the strength of those bonds that I found genuinely fascinating, because I always assumed it was just like a zipper, totally uniform all the way up and down, but it's really not.
It's not uniform at all. The connection between G and C is actually significantly stronger. It's held together by 3 hydrogen bonds. The connection between A and T only has 2.
So G and C is like a heavy-duty industrial weld, and A and T is more like a lighter glue.
That's it, exactly. And this actually has real-world, life-or-death evolutionary consequences. If you are a bacterium living in a boiling hot spring in Yellowstone National Park, you are going to evolve to have a genome heavily packed with GC pairs.
Because you don't want your DNA to physically melt in the hot water.
Precisely. If your DNA was mostly AT pairs, the environmental heat would just unzip your double helix, your code would fall apart, and you'd die. So the extreme environment literally dictates the chemical composition of the genetic code.
That is wild to think about. Now, there's another structural thing in the material that always confuses me a bit. The anti-parallel strands. The text keeps talking about 5' and 3'. What does that actually mean in plain English?
It refers to the specific carbon atoms in the sugar ring of the backbone. The sugar molecule is shaped like a pentagon. Chemists number those 5 corners 1 through 5. The phosphate group is attached to the 5th carbon, the 5' end, and the hydroxyl group, which is the connector piece, is on the 3rd carbon.
Okay, so it's really just orientation, like calling the ends head and tail.
Right, but here is the major catch. The 2 strands of the DNA double helix run in completely opposite directions. One strand runs 5' to 3', let's call that the northbound lane. The opposite strand runs 3' to 5'. That's southbound.
Like a divided highway.
Exactly like a divided highway. And this matters immensely because the special enzymes that copy DNA, the cellular construction crew, they can only drive in one direction. They can only ever build new DNA from the 5' end toward the 3' end.
I can easily see how that would cause a massive traffic jam.
It causes a huge headache during the replication process, which we will definitely get to. But before the cell even copies the DNA, we have a much more basic problem, a storage problem. The source notes that a single E. coli chromosome, if you were to somehow stretch it out in a straight line, is about 1 millimeter long.
Which sounds really small to us.
But the bacterial cell itself is only about 1 micrometer long. That is a 1,000-fold difference in size.
That's like trying to stuff an entire kilometer of thick rope into a tiny shoebox.
Or trying to pack for a month-long vacation using a tiny carry-on bag. How do you fit it all in?
You just sit on the suitcase and pull the zipper as hard as you can.
Basically, yes.
Yeah.
Bacteria use a very specific physical process called supercoiling. They don't just shove the DNA in randomly, they actively twist it. Imagine holding a thick rubber band. If you hold one end perfectly still and start twisting the other end, the rubber band starts to knot up and fold in on itself. It becomes much tighter and highly compact.
So it coils up into this dense little ball.
Yes. And they have specialized enzymes called topoisomerases that do all this heavy lifting. They actively wind up and twist the DNA so it neatly fits inside the tiny volume of the cell.
Now, this DNA sits directly in the cell fluid, but there's a big fundamental difference between how bacteria, the prokaryotes, store their DNA and how we eukaryotes do it. We have a protective nucleus, they don't. But the text also points out something else, the backup copy issue.
This is fundamental for understanding why bacteria can evolve so incredibly fast. We eukaryotes are generally diploid. We have 2 copies of every single chromosome, one from mom, one from dad.
So if I happen to have a broken or bad gene on one copy, the other one can usually cover for it.
Right. Recessive traits can just hide in the background. You have a built-in genetic backup system. Bacteria, however, are haploid. They just have a single circular chromosome, one single copy of the manual.
So if a mutation happens,
Boom. It is expressed immediately. There's nowhere for a bad gene to hide. If that sudden mutation is bad, the bacteria just dies instantly. But if it's good, it thrives instantly.
it's like playing high-stakes roulette with your entire existence.
It absolutely is. Every single change is instantly tested by the harsh environment. That is exactly why you see things like antibiotic resistance emerge so rapidly in hospitals. They aren't carrying around silent recessive resistance genes for generations waiting for the right moment. It's immediate trial and error.
But they do have these little genetic expansion packs we briefly mentioned earlier, the plasmids.
I love plasmids. These are these small, separate, secondary loops of DNA that just float around outside the main chromosome. They aren't strictly necessary for the bacteria to live its day-to-day life. They don't run the basic housekeeping functions, but they carry special superpowers.
Like downloadable content for a video game, the DLC.
Exactly. The main bacterial chromosome is just the base operating system. The plasmid is the DLC that you install to get invincibility against penicillin or the ability to digest nylon. And the really scary part is bacteria can swap these plasmids with each other like kids trading Pokemon cards.
We will definitely get to that swapping in a bit. But first, we have the machine built, we have the code stored. Now we actually need to run the program. This brings us to section 2, the flow of information, the central dogma of biology.
DNA to RNA to protein.
It all starts with replication, copying the hard drive. The text explicitly calls this semi-conservative replication. Why that specific term?
Because you don't just throw away the old piece of DNA and build 2 brand new strands from scratch. You unzip the existing double helix down the middle, and each of those old strands acts as a physical template to build a new one.
So every new DNA molecule is exactly half old material and half new material.
Exactly. It's semi-conservative. You conserve half of the original.
And the physical location where this unzipping happens is called the replication fork. I kind of imagine this looking like the zipper on a heavy winter jacket.
That's a really good visual. But imagine the zipper is stuck, and you really have to force it open. And imagine the fabric of the jacket is actually made of heavy rope that is tightly twisted together. As you aggressively pull that zipper up, the twisted rope above the zipper gets tighter and tighter and tighter.
Oh, right, because it's a helix. If you pull the 2 strands apart, that built-in twist has to go somewhere.
It creates immense physical tension just ahead of the fork. If you don't constantly relieve that building tension, the DNA molecule will literally snap under the pressure and the cell dies.
So what does the cell do to stop that?
It brings in those topoisomerase enzymes we talked about again, specifically one called DNA gyrase. It constantly sits just ahead of the moving fork. It temporarily cuts the DNA strand, lets it untwist just a little bit to relieve the pressure, and then perfectly glues it back together.
It's basically a highly specialized stress reliever enzyme.
Literally. It keeps the road clear so the replication machinery doesn't derail.
So once we've safely copied the code, we actually need to read it to do something useful. That's transcription, turning DNA into RNA.
Think of the DNA as the master reference book in a massive library. It's rare, it's highly valuable, and you are absolutely not allowed to take it out of the reference section. But you desperately need the specific instructions on page 42 to build a table.
So I just walk over and photocopy the page I need.
That temporary photocopy is the mRNA messenger RNA. And the enzyme called RNA polymerase is the photocopier. It reads the master DNA and spits out a cheap, single-stranded RNA copy.
And there are some chemical differences here. The text notes that RNA uses a letter called uracil instead of thymine.
Yes, it swaps T for U. And it uses a slightly different sugar molecule in its backbone. But the main functional difference is that it's single-stranded and it's highly unstable.
Unstable? Doesn't that mean it falls apart? Isn't that bad?
No, it's actually a brilliant feature. You don't want these instruction manuals floating around the cell forever. You want to build the protein you need right now and then immediately shred the instructions so you don't accidentally make too much and waste resources. The instability allows the cell to constantly change its mind.
Okay, so we have our temporary mRNA blueprint. Now we have to take it to the construction site. The ribosome. This step is translation.
The ribosome is a complex molecular machine. In bacteria, it's called the 70S ribosome. It physically clamps onto the mRNA strand and starts reading it.
This is where we get into the actual language of life. The genetic code, the codons.
Right, the basic language of DNA only has 4 letters. But the proteins we need to build are made of amino acids, and there are 20 different amino acids. So you need a translation dictionary to get from 4 to 20. That's what a codon is. 3 letters of RNA equal 1 specific amino acid.
So the sequence AUG means start here?
And it also physically places an amino acid called methionine at that starting spot. The sequence GCC might mean alanine. The ribosome simply reads these 3-letter triplets, and another little molecule, tRNA transfer RNA, acts as a tiny delivery truck.
The tRNA brings the specific molecular brick that matches the order on the blueprint.
Exactly. The ribosome takes those bricks and links them together one by one to form a long protein chain.
And here's where the bacteria really show off their speed again. I found this detail completely incredible. In human cells, this whole process happens in totally different rooms. The DNA is locked in the nucleus, the ribosomes are out in the cytoplasm, but bacteria are essentially just a tiny studio apartment.
There's no nucleus, there are no walls, everything is happening in the exact same room. So as the RNA polymerase is still in the process of ripping the mRNA copy off the DNA, literally while the ink is still wet on the photocopy, a ribosome can just jump onto the start of that mRNA strand and immediately begin translating it.
It's doing both processes at the exact same time.
Yes. It's a phenomenon called coupled transcription and translation. It's like reading a stock ticker tape while it's still printing out of the machine.
That perfectly explains why they can react to their environment so incredibly fast.
It's near instant. If a bacterium suddenly senses a new food source like sugar, it can start pumping out the specific digestive enzymes to eat it within seconds. It doesn't have to wait for the photocopy to be completely finished, packaged, and mailed to another room.
Speaking of reacting to the environment, this leads us nicely right into Section 3, smart regulation. Because bacteria don't just run every single program all the time, right? That would be exhausting and wasteful.
It would be incredibly wasteful.
Yeah.
If you are a bacterium swimming in a giant pool of glucose, why would you waste precious cellular energy-building enzymes to digest lactose? You simply wouldn't. Bacteria are ruthless, efficient economists.
The text highlights this model called the operon. This is essentially the control switch.
Right. An operon is a cluster of related genes that are all controlled by one single master switch. It's exactly like a power strip in your office. You flip one physical switch, and your computer, your monitor, and your printer all turn on at the same time.
The classic textbook example is the lac operon. This handles lactose digestion, and this is described as an inducible system.
Think of the lac operon as a standard light switch that is normally in the OFF position.
Okay, so normally the room is dark, nothing is happening.
Yes. Normally, a specific protein called a repressor is physically sitting right on the DNA strand, completely blocking the path of the RNA polymerase. I always like to think of this repressor as a massive bouncer standing in front of the door to an exclusive club. He's big, he's grumpy, and he is absolutely not letting the polymerase in.
So the genes don't get copied. 0 lactose enzymes are made.
Correct. But then, you drink a big glass of milk. Lactose suddenly enters the bacterial cell, specifically a modified form called allolactose. This lactose acts as the inducer. It binds directly to the repressor, the bouncer.
It's a bribe.
It is exactly a bribe. When the lactose binds to the repressor physically changes its shape. Because of that new shape, it can no longer grip onto the DNA. The bouncer literally falls off the door.
And the door is suddenly wide open.
The RNA polymerase zooms right in, transcribes the genes, the cell immediately makes the enzymes, and all that new lactose gets digested.
And what happens once the lactose is all gone?
The bribe is gone. The bouncer just reverts back to his original grumpy shape, sits right back on the DNA, and shuts the whole system down again. It's beautifully simple, purely demand-based manufacturing.
But there is a catch here, a secondary level of control. Because even if lactose is sitting right there, if glucose is also present, the cell just completely ignores the lactose.
This is a mechanism called catabolite repression. It answers a very basic priority question. What if I have a premium steak, which is glucose, and a piece of stale bread, which is lactose? You're going to eat the steak first every time.
So how does the cell actually know which one is available?
It uses an internal alarm signal molecule called CAMP. Think of CAMP as a cellular hunger signal. When glucose levels are high, the cell is full and happy. CAMP levels stay very low.
Okay, makes sense.
But when glucose finally runs out, the cell starts starving, it gets hungry, so CAMP levels rise rapidly. This CAMP molecule binds to a helper protein called CAP. And this CAMP protein binds to the DNA and actively pushes the RNA polymerase forward.
It's like a volume dial.
Exactly. The bouncer falling off just unlocks the door. But the KKP protein is the guy who actually shoves the door open and gets things moving.
Yeah.
If you have lactose, so the door is unlocked, but you still have high glucose, so there's no shove, the door basically stays closed.
Yeah.
You need both conditions met perfectly. Lactose must be present, and a glucose must be absent.
That is surprisingly complex Boolean logic for a single microscopic cell.
It really is. If lactose and a not glucose, then transcribe.
Amazing. Now let's flip it around. The trip operon. This controls making tryptophan, which is an amino acid. The text calls this a repressible system.
Right. So the lac operon was a make it only when you need to eat it system. The trep operon is a make it constantly until you have enough system. Think of it more like a thermostat and a furnace in your house.
Okay. The furnace is normally on N to keep the house warm in winter.
The trep operon is normally on N because the cell constantly needs a supply of tryptophan to build its proteins. But if it suddenly gets really hot outside, say you find yourself swimming in a tryptophan-rich environment, you don't need to run the furnace anymore.
So the final product itself reaches up and turns off the switch.
Yes. The abundant tryptophan acts as a co-repressor. It binds to the trap repressor and activates it. It essentially hands the repressor the key to clamp onto the DNA and halt all further production.
It's a classic feedback loop.
Negative feedback. Too much of the final product physically shuts down the factory.
Now, there is another deeply complex layer of control in the trep operon called attenuation, and honestly, reading this specific part gave me a bit of a headache. It involves the RNA strands spontaneously folding into loops.
Attenuation it's functionally beautiful, but I agree, it's easily one of the hardest concepts in the entire chapter. It relies entirely on that coupled transcription and translation speed hack we talked about earlier.
Where they happen in the same room at the same time.
Right. The cell essentially uses the fast-moving ribosome as a real-time sensor. At the very beginning of the mRNA strand, section called the leader sequence, there's a little built-in test. It has two tryptophan codons sitting right next to each other.
So the ribosome has to actually find two physical tryptophan molecules to pass the test and keep moving.
Exactly. If the cell is genuinely starving for tryptophan, the ribosome gets to that exact point and just stalls out. It's waiting for parts to arrive. It's like a factory worker saying, hey, where's the tryptophan I need?
And what happens to the RNA while it waits?
Because the ribosome is stalled, the long mRNA tail trailing behind it naturally folds into a specific shape, a stem loop structure that effectively says, keep going, we need more. It physically signals the polymerase up ahead to continue making the rest of the manufacturing genes.
But what if there is plenty of tryptophan already?
Then the ribosome zooms right through that little test. It doesn't pause for a microsecond, and because it moves so fast, it physically blocks that first loop from forming. Instead, the mRNA folds into a completely different shape further down, a terminator loop.
A terminator loop. That sounds aggressive.
It is. This terminator loop is so bulky, it physically yanks the RNA polymerase right off the DNA track. It abruptly snaps the chain. It effectively says, we have plenty of tryptophan, abort the process, stop wasting energy.
So it's literally a tripwire. If you run through it too fast, you trip the wire and accidentally shut down the entire factory.
Precisely. It allows the cell to perfectly fine-tune its production based on the exact second-by-second levels of amino acids floating around.
That is just mechanical genius. So that's how the incredible machine runs when things are going well, but machines inevitably break. Or they get hacked. Which brings us to section 4. The remix. Mutations.
Evolution absolutely requires change. If DNA was perfectly stable and never ever changed, life on Earth would still just be a puddle of slime mold. Mutations are the raw engine of change.
The text first mentions spontaneous mutations. These are basically just random typos.
Just typos. The polymerase copying enzyme is incredibly good at its job, but it's not perfect. It makes a mistake about once in every billion bases it reads.
That sounds incredibly rare, though.
It is rare for one single cell. But when you have a massive colony of billions of bacteria, and they're all replicating every 20 minutes, that rare typo happens all the time across the population.
Then you have induced mutations caused by mutagens.
The sunlight. Ultraviolet radiation is a massive one. UV light physically hits the DNA strand and causes two adjacent thymine bases to chemically fuse together. They form what's called a thymine dimer.
It's like two adjacent bricks in a wall suddenly melting together into one weird block.
And when the polymerase enzyme cruises along and tries to read that melted block, it hits a massive speed bump. It can't read it. This usually causes severe copying errors or just stops replication dead in its tracks.
Now, let's talk about the actual severity of these typos. A base substitution is just swapping one single letter.
That's a point mutation.
Yeah.
Say you replace an A with a G. It might change one single amino acid in the final protein. Maybe the protein works slightly better, maybe slightly worse, or maybe it doesn't affect the shape at all and it doesn't matter.
But a frameshift mutation sounds pretty disastrous.
A frameshift is when you accidentally add an extra letter or delete one entirely. Remember, the genetic code is strictly read in blocks of 3. Think of a sentence like, the cat ate the rat.
Right, all 3-letter words.
If I accidentally delete the C in CAT, the rigid reading frame shifts over by one letter. The sentence becomes the atai et, he, her, at.
It just turns into complete gibberish.
Total, unreadable gibberish. Every single amino acid placed after that deletion point is going to be wrong. The final protein is usually completely destroyed and non-functional. Frame shifts are almost always lethal to that specific gene's function.
And when the physical damage to the DNA is really bad, like from massive, prolonged UV exposure, the bacteria enter a panic mode called the SOS response.
This is the ultimate Hail Mary pass. The DNA is so severely damaged that normal replication just completely stops. The cell realizes it is about to die, so it activates the extreme SOS system. It produces a special emergency polymerase that is, let's just say, very loose with the rules.
It's a sloppy, desperate builder.
Extremely sloppy. It doesn't care at all about perfectly matching A to T or G to C. It just rapidly jams absolutely anything into the gaps just to bridge the broken DNA strands so replication can finally finish.
So it completely sacrifices accuracy just for a chance at survival.
Yes. It creates a massive wave of rapid mutations. Most of those mutations might be terrible, but the cell is desperately gambling that at least one of them might somehow allow it to survive whatever stress is killing it. It's evolution forced into overdrive.
Now, besides internal typos, bacteria have another huge way to change their code: bacterial sex, or more accurately, horizontal gene transfer.
I definitely prefer horizontal gene transfer because it's not reproduction in the way we think of it. It's sharing. Humans rely on vertical transfer parents passing genes down to children. Bacteria utilize horizontal transfer passing genes sideways from neighbor to neighbor.
There are 3 main ways this happens according to the source. First up is transformation, and this circles us all the way back to Griffith and those dead mice.
Right. Transformation is a bacterium just picking up naked DNA from its surrounding environment. A bacterium dies, its cell wall bursts open, and its internal DNA spills out into the fluid. Another completely different bacterium swims by, chemically senses it, says, hey, that looks like useful code, and just pulls it inside.
But you have to be in a state called competent to do this.
Yes. Not all bacteria can just pull in naked DNA all the time. They usually only become competent when they are highly stressed or starving. It's a desperation survival mechanism. Basically thinking, I'm dying anyway, maybe this dead guy had a better genetic idea.
The second way is conjugation, the mating bridge.
This is definitely the most deliberate method of transfer. You have a donor cell, called the F+ cell, meaning it's fertile. It contains a specific plasmid. It actively builds a physical, hollow tube called a pilus.
It sounds like a grappling hook.
It functions exactly like one. It shoots out, lassos the recipient cell, reels it in close, and physically connects their cytoplasm. Then it unschools a copy of its plasmid and shoots it straight through the tube into the neighbor.
And this right here is exactly how antibiotic resistance spreads so terrifyingly fast in hospital wards.
Exactly. One single bacterium figures out how to beat penicillin, and it effectively just emails the exact blueprints to every other bacteria in the room. It forms a rapid peer-to-peer network of resistance.
And there's a weird, extreme version of this involving HFR cells.
High frequency of recombination. This crazy scenario happens when that transfer plasmid doesn't just stay separate, but actually integrates itself directly into the bacteria's main chromosome. When this specific cell tries to mate, it doesn't just send the plasmid, it tries to push its entire massive chromosome through that tiny tube.
Yeah, it tries to send the whole library.
Yes. It almost rarely finishes because the delicate tube inevitably breaks from environmental movement, but it usually ends up transferring a huge chunk of its own core DNA to the recipient. It's a major driver of large-scale genetic mixing in the wild.
The third way is transduction, and this one involves a totally unaware third party, a virus.
A bacteriophage. This is a specific type of virus that only infects bacteria.
It's essentially a packaging mistake, right?
It is a total biological accident. The virus is busy reproducing inside the doomed bacteria, chopping up the host DNA and packaging its own viral DNA into fresh new viral shells. But sometimes it makes a mistake and accidentally grabs a random piece of the chopped up bacterial DNA instead.
Yeah. A completely careless packaging error.
Exactly. Then that newly minted virus bursts out and goes to infect a fresh new bacterium. But instead of injecting deadly viral DNA to kill it, it accidentally injects the harmless bacterial DNA it stole from the previous victim. It acts as an accidental syringe, carrying genetic information from one cell to another.
Finally, in the remix section, we have transposons, jumping genes.
Barbara McClintock originally discovered these in corn plants, but they play a huge role in bacteria. These are unique segments of DNA that contain their own built-in scissors, an enzyme called transposase.
So they can literally cut themselves out of the code?
They cut themselves entirely out of one spot in the genome and physically paste themselves into another random location.
They sound like complete chaos agents.
Total chaos. If they happen to land right in the middle of a vital, life-saving gene, they completely break it. But the real danger is they can also jump from the main chromosome onto a mobile plasmid.
And since we know plasmids love to travel.
Exactly. A transposon can pick up an antibiotic resistance gene, snip itself out, jump onto a mobile plasmid. The plasmid then moves to a completely new bacterial species, and the resistance spreads across species lines. They're the ultimate vehicles for rapidly moving genes between different DNA molecules.
This neatly brings us to section 5. The viral connection. We just briefly touched on bacteriophages, but these things are visually fascinating. They literally look like the Apollo lunar landers.
They really do. They have this geometric icosahedral head or rigid tail sheath and little spindly legs called tail fibers. They land precisely on the surface of the bacterium, punch a microscopic hole through the armor, and forcefully inject their DNA inside like a hypodermic needle.
The outline details 2 distinct life cycles here. The lytic cycle is basically the pirate takeover.
That's your standard aggressive viral attack. The virus injects its DNA, completely hijacks the host's cellular machinery, forces it to manufacture 100 copies of the virus, and then violently explodes the cell, which is called lysis, just to let the new viruses out. It's violent and it's fast.
But the lysogenic cycle is much more subtle. It's the sleeper agent approach.
This is the truly creepy one. The viral DNA enters the cell, but it doesn't immediately kill it. Instead, it quietly integrates itself right into the host's bacterial chromosome. It becomes what we call a prophage.
It just goes deep undercover.
It sits there totally quietly, and every single time the bacterium naturally replicates its own DNA, it flawlessly copies the hidden viral DNA right along with it. The virus is effectively hitching a free ride through countless generations of bacteria.
But what is it waiting for?
It's waiting for the ship to start sinking. If the host bacterium suddenly gets stress-hit by UV light, exposed to toxic chemicals, the hidden virus chemically senses that its host is in serious trouble. It immediately pops itself out of the chromosome, violently switches back to the lytic cycle, makes 100 copies, and kills the cell to escape before the ship goes down.
But while it's just sitting there undercover, it can do this weird thing called phage conversion. It actually changes the bacteria's personality.
This is a shockingly crucial medical point. Take Corynebacterium diphtheria, the bug that causes the deadly disease diphtheria. By itself, it is basically harmless.
Wait, really? The disease is harmless?
The bacteria is harmless. It only actually causes the deadly disease if that specific bacteria is actively infected by a specific lysogenic virus. The actual genetic code for the deadly diphtheria toxin is located on the viral DNA, not the bacterial DNA.
So when a human gets diphtheria, we're actually getting sick from a germ that is itself sick with a virus.
Exactly. The exact same thing is true for cholera. The devastating cholera toxin is entirely viral in origin. It really forces us to redefine what a human pathogen actually is.
That is just mind-bending. Okay, section 6. We've looked deeply at how nature runs this machine. Now, how do we hijack it? Genetic engineering. The source clearly states this entire modern field was born when scientists discovered restriction enzymes.
This is the absolute foundation of biotechnology. Restriction enzymes are nature's microscopic scissors. Bacteria originally evolved them millions of years ago as a primary defense system against those exact viruses we just talked about.
So they use them to chop up the invading viral DNA.
Yes. If a virus injects its DNA, the restriction enzyme rapidly scans the incoming code. If it finds a very specific target sequence, it ruthlessly cuts it. Game over for the invading virus.
But these scissors leave a very specific type of cut. The notes call them sticky ends.
This is the magical key to all genetic engineering. The enzyme doesn't just cut straight across the double helix. It cuts in a jagged, staggered line. Let's say the enzyme looks for the sequence GATTC. It cuts between the G and the A on both opposing strands.
So you are left with a little single-stranded overhang on each side.
A single-stranded tail. And because of how DNA pairs up, that tail desperately wants to find and pair with a perfectly matching tail. It acts exactly like microscopic Velcro.
So if I take a human gene and cut it with this enzyme, and then I take a bacterial plasmid and cut it with the exact same enzyme.
They will both have perfectly matching sticky ends. You mix them together in a test tube, and they naturally find each other and stick. Then you just use one more enzyme, called DNA ligase, to permanently glue the backbone shut.
And voila! You have successfully created a bacterial plasmid carrying a human gene, a vector.
Exactly. You then transform that engineered plasmid back into a normal E. coli bacterium, and suddenly that tiny bacterium is actively reading the human gene and producing complex human protein.
But DNA is completely invisible to the naked eye. How do scientists actually know if this tiny cut-and-paste job worked? The outline mentions a technique called gel electrophoresis.
This is exactly how we see what we've done in the dark. You take your manipulated DNA sample and inject it into a flat slab of agarose gel. It physically feels a lot like very firm jello. Then you apply a strong electric current across the gel.
And because DNA is naturally negatively charged, it gets physically pulled toward the positive side of the gel.
Right. But the dense gel acts like a microscopic sieve or a complex maze. The really small pieces of cut-up DNA can easily zip through the tiny holes and move very quickly across the gel. The large, bulky pieces get tangled up in the matrix and move much slower.
So it's basically a highly controlled foot race.
It's exactly a race. Over a couple of hours, the invisible DNA separates into distinct, visible bands based entirely on their physical size. You can then look at the gel under a light and say, aha, there's my specific gene. It traveled the exact right distance, so it's exactly the right size.
And what if you need to find one very specific needle-in-a-haystack sequence in that massive mess of bands? That's southern blotting.
Southern blotting is basically playing Where's Waldo with DNA. You transfer all those separated DNA bands from the fragile gel onto a sturdy piece of special paper. Then you literally wash the paper with a radioactive probe.
A radioactive probe.
It's just a very short, synthesized piece of single-stranded DNA that is custom designed to stick only to the specific target gene you are looking for. You wash it over the paper, and it completely ignores everything else and sticks it right to your target. Then you hold a piece of X-ray film up to the paper, and that exact spot glows black on the film.
Waldo is radioactive, so he's easy to spot.
Exactly. It completely eliminates the guesswork.
And finally, actually reading the raw code itself, sequencing. The outline highlights the Sanger method.
This is pure, elegant chemical genius. You essentially run a standard DNA replication reaction in a test tube, but you intentionally add in special, modified poison nucleotides. They're called D-deoxynucleotides.
Poison, how does that help you read it?
Because when the copying enzyme inevitably grabs one of these fake poison letters and adds it to the chain, it physically can't add another one after it. The chain extension stops permanently right at that spot.
So if you have a tube full of poison A letters, every single time the machine tries to write an A, there's a random chance it grabs the poison one and stops the whole process.
Yes. You run 4 separate reactions in 4 tubes. 1 tube for A, 1 for T, 1 for G, and 1 for C. You end up with millions of copied fragments of all different lengths, but you know every fragment in the A tube strictly ends with an A. By sorting all those millions of fragments by exact size on a huge gel, you can actually read the precise sequence, letter by letter, from the bottom to the top.
And that incredibly tedious method is how we originally sequenced the entire human genome.
That was the foundational method, yes. We have vastly faster automated laser ways to do it now, but Sanger's logic was the ultimate breakthrough that made reading the code possible.
Which brings us nicely to Section 7. Applications and ethics. We have all these incredible tools. What have we actually done with them?
The absolute biggest and clearest win is in human medicine. Before genetic engineering became viable, if you were diabetic and needed life-saving insulin, we had to literally harvest it from the crushed pancreases of slaughtered pigs and cows.
That sounds incredibly inefficient and messy.
And it wasn't perfect. Animal insulin is slightly different, and some people develop severe allergic reactions to the pig proteins. Now, we just take the pure human insulin gene, paste it into bacteria or yeast cells, and they tirelessly pump out pristine, identical human insulin in giant industrial vats.
It's vastly cleaner, infinitely safer, and much cheaper.
The exact same logic applies to the hepatitis B vaccine. We used to actually have to extract the virus from the blood plasma of heavily infected patients to make the vaccine. Extremely dangerous work. Now we just safely clone the gene for the viral surface protein into harmless yeast. The yeast manufactures the protein, we purify it and inject it. You get full immunity without anyone ever touching the actual dangerous virus.
Then there's this concept called site-directed mutagenesis. This sounds more like hardcore research than manufacturing.
This is where scientists really get to play editor. Instead of just waiting around for random mutations to happen and hoping for something interesting, we intentionally go in and change one single specific letter in a gene to see what happens. We ask, what exactly happens to this enzyme's function if I deliberately change this one alanine to a glycine?
It's like deliberately introducing typos in a spell checker just to see how the meaning of the sentence changes.
Exactly. It's how we slowly reverse engineer and truly understand exactly how these complex proteins fold and work on an atomic level.
But looking at the source material, it's clearly not all been smooth sailing and purely saving lives. The source explicitly brings up the ice- bacteria controversy. I had honestly never heard of this before.
Oh, this was a massive public event in the 1980s. There is a completely natural bacterium called Pseudomonas syringae that lives all over agricultural plants. It naturally manufactures a specific surface protein that acts as a nucleation point, which actively helps ice crystals form at slightly higher temperatures.
So it's directly responsible for causing devastating frost damage on farmers' crops.
Yes. So genetic scientists had a very logical idea. Let's just go in and neatly delete that one specific ice-forming gene. They successfully created the engineered ice- strain. The brilliant idea was to spray this harmless bacteria onto strawberry fields to protect them from sudden frost snaps.
Sounds like an absolute miracle product for farmers.
But the general public completely panicked. This was basically one of the very first authorized releases of a genetically modified organism, a GMO, into the open wild environment. People understandably asked, what if this weird bacteria permanently changes the global weather patterns? What if this artificial deletion jumps into other bacteria and causes unknown ecological havoc?
And did any of that happen?
No, it was perfectly safe. But the sheer public outcry absolutely ignited the massive ongoing ethical and legal debate we are still having today about releasing GMOs into the food supply and the wild.
And finally, in this section, RFLP, DNA fingerprinting.
Restriction fragment length polymorphism. It is a terrible mouthful of a name. But the concept is beautiful. It basically means your DNA sequence and my DNA sequence are extremely similar, but slightly different. If I cut your specific DNA with E-core-R1 scissors, and I cut my DNA with the exact same E-core-R1 scissors, the physical cuts will happen in slightly different places because our base letters are different.
So when we run that gel race we talked about, my pattern of bands will look completely different from your pattern.
Exactly. It creates a completely unique, unforgeable genetic barcode for every individual. This is exactly how we conclusively catch criminals at crime scenes, how we positively identify paternity, or how epidemiologists track a deadly salmonella outbreak back to one specific processing factory.
It's the absolute backbone of forensic science.
And it is built entirely on the backs of microscopic bacterial defense enzymes.
So stepping back, we've gone from looking at a static, boring diagram of a double helix in a textbook to this wildly dynamic, constantly jumping, swapping, viral infected world of microbial genetics.
It really fundamentally changes your perspective on biology. We as humans tend to think of life and genetics as this very stable, locked-in thing. But at the microbial level, it is incredibly fluid. Genes are basically just commodities. They're actively traded, stolen, and hacked constantly to survive.
And the big picture takeaway here from all this material.
These seemingly invisible mechanisms, the transcription, the translation, the constant recombination, they are the literal foundation of all living things on Earth. But more importantly for us right now, truly understanding them has handed humanity the keys to the kingdom. The entire modern biotechnology revolution is just humans finally learning to fluently speak the chemical language that bacteria have been speaking to each other for 3.5 billion years.
And that leads me to a really provocative final thought to leave you with. Bacteria have been extensively genetically engineering themselves via these mobile plasmids and transposons forever. They are the original master biohackers. Humans have really only just joined the game in the last few decades. As we get better and better at writing this code, designing totally new proteins, targeting new vaccines, maybe even coding entirely new synthetic life forms from scratch, are we just cleverly copying nature's old tricks? Or are we finally crossing a threshold into creating something entirely unprecedentedly new?
That is the ultimate question of our era. We are rapidly transitioning from merely being passive readers of the genetic code to active writers of the code. And the core syntax we are using to write the future is entirely bacterial.
Definitely something to chew on the next time you casually squirt some hand sanitizer on your palms. You're not just killing a few germs, you are wiping out a sprawling, ancient supercomputer. Thanks for joining us on this deep dive into the invisible code.
Always a pleasure to decode it with you. Keep asking questions.
See you next time.